I would like to introduce you all with our next chair, Dr. Tamanna Narsinghani, and our keynote speaker, Dr. Pia Vogels, to our esteemed audience. Ma'am, please. Now, I would like to request Dr. Rizwan Khan, Principal Skyper, to greet them with a bouquet. Dr. Tamanna Narsinghani is working as Associate Professor at School of Pharmacy, Devi Ahilya Vishwavidyalaya, Indore. Dr. Tamanna has more than 19 years of teaching experience. She has published several research and review papers in journals of national and international repute, published one Indian patent, presented research papers in various national and international conferences, and authorized 5 books. She is MPharm, Gold Medalist and recipient of AICTE Junior Research Fellowship. She qualified GATE with 97.97 percentile and 69th All India rank. She was awarded Certificate of Appreciation in 2017 for getting the highest citations of research publications during 2012 to 2016 among university readers. She also received Special Appreciation Award for poster presentation at International Conference on Emerging Challenges in Biotechnology, Human Health and Environment, and 8th Annual Convention of Association of Biotechnology and Pharmacy in 2014. She is actively involved in documentation of NAC, IQAC, NIRF ranking of Devi Ahilya University. Now, I would like to request Dr. Tamanna to throw light on the topic for and invite Dr. Pia Vogels.
Good morning, all of you. It gives me immense pleasure to introduce our eminent speaker for this session, Dr. Pia Vogel. Professor Pia Vogel is a biological sciences professor at SMU. She is a biochemist, leading authority in electron spin resonance spectroscopy. Professor Pia Vogel's research trends include structure, molecular dynamics, protein-protein interactions of nucleotide binding proteins. At SMU, she is the founding director of the Center for Drug Discovery, Design and Delivery in CD4. She has received $1.5 million as a grant to support her research, including major grants from the NSF and the NIH. She also holds an honorary degree from the Shaanxi University in Taiwan, China. Her topic for today's speech is overcoming multi-drug resistance in cancer. We are highly motivated and having keen interest in listening to you, ma'am. Please, you are kindly invited for a minute talk. Thanks a lot.
I'm so honored to be here with all of you, and I can't thank the organizers and the helpers and the committee enough to have me speak to you today. So we're going to take a big step back, okay? We're not even animal models. We are at the very beginning of trying to develop some potentially new drugs. And actually, Dr. Tiwari has made it easy for me to get to introduce it, because he more or less introduced my problem or my research idea. So, as was said earlier, I'm a protein biochemist, so I was originally interested in trying to understand how proteins work. And then at some point, a protein that is important for medicinal reasons, and I want to be careful of the time, became interesting to me, and that is a protein that is associated with multi-drug resistance in cancers. So there's a lot of different ways, and Dr. Tiwari has talked about that yesterday, how cancer cells can become resistant to chemotherapy. The one mechanism that we are interested in is the one that is an ATP-dependent toxin pump that gets overexpressed, especially in metastatic cancers or cancers that have undergone chemotherapy and have recurred. So let me go forward here. Okay, so as soon as those pumps are overexpressed, the drugs get pumped out. You don't get enough therapeutic concentrations, and the cells can survive. So what we're hoping is to find a way on how to block these pumps with small molecules. We don't want to really downregulate the synthesis of the pumps because they have evolved to be helpful for the body by reducing toxins. So the two proteins we're studying currently are both they're siblings almost, or cousins. They're very closely related. It's the P-glycoprotein and the BCRP, the breast cancer resistance protein. They are membrane proteins who use the cellular energy currency ATP. They're found in all kinds of different tissues. They're also important in the blood-brain barrier. So they use, as I said, they use ATP to pump toxins out of and waste products out of cells. So they're important for us. But chemotherapy, those are toxins, right? So they have evolved to pump a wide variety of chemically very unrelated substances out of cells. Because they are so generic in what they can pump, it makes it also very hard to somehow come up with a design for a pump inhibitor. And in fact, people have been looking for a very long time, because we know for 40-something years that these pumps are problematic in chemotherapy-resistant cancer. So we have thought maybe we can find a way to very specifically inhibit the pump, not by trying to, inhibit this very plastic transport portion of it that has evolved to be very multifunctional, but to inhibit what's very specifically needed to hydrolyze ATP. So for this we needed structures, and we needed to understand the mechanism of the pump. At the time we had crystal structural models of 4 different related proteins. So those are those are not human. The one furthest to the left is from mouse, and the others are all bacterial. They are actually descend with modification. That's evolution. So nature has at some point discovered a good mechanism, and that's been used over and over. So we had these 4 different structures, and they kind of looked as if they could represent the proteins in different stages of its catalytic turnover, right? So the one to the left is open to the inside of the cell. These are the nucleotide binding domains. This is the drug binding domain. This down here is turned around, okay? So a little bit, so as long as they are open to the inside, they're close to the outside. And then if we go on and continue to close those nucleotide binding domains, getting into a structure where ATP can actually be hydrolyzed, it results in the opening of the drug binding domain to the outside of the cell, and the drugs can get exported. Well, but it was not a human protein that we had the structure of, so we had to get a human structural model. And I just want to make sure that you understand that we understand that these are models, right? These are not real structures. These are just hypothesis generating ideas of what it could potentially look like. So we started with the known structure of the homologous proteins that I showed you before, and we had a known sequence of the human protein. So we just built the human protein onto the human sequence onto the known structure, tested for problems, and hey, there was a structure that looked like the bacterial pump but had the human sequence in it. So of course we tested that a little bit, and we thought, well, we had those, different structures available. Can we, in the computer again, make a simulation that pushes one structure into the other, and can we model the catalytic turnover that way? And indeed we can. Okay, so this actually, yeah. So we can recreate by just computationally how those nucleotide binding domains come together, and then at the point where ATP gets hydrolyzed, the drug binding domain opens to the outside. So the next question now is, so again, it's just opportunities for generating hypotheses, okay? So the next question was, what happens if we add a transport substrate to it, okay? For that we used another tool, and that is in silico drug docking, okay? Those are programs that are available. They have limitations, obviously, but again, they could give us a hypothesis and then we can test it. So we went on and used at first some known transport substrates. So in the process, the program kind of takes a random drug conformation, throws it at the protein, and then sees where it binds, calculates the delta G of binding, modifies the conformation, throws it out again. So this is a repeated process over time, and then we get in the end some best binding interaction, and we can correlate that with published results, because there is a lot of biochemical research that has happened here. So it actually fit quite well. When we docked daunorubicin or verapamil, those are two very well studied transport substrates of the pump. They actually interacted in our model just the same way that it had been reported in biochemical studies. So we said, okay, let's just see what happens if we put the, this is I think daunorubicin, if we dock it where it docks in the structure, and then just see what happens when we run through the catalytic cycle, and you can see that it opens, it's pumped through the membrane, and it's open down here, and it can dissociate from the pump. So a nice little way of visualizing on how that pump may actually work. They bring things through. So how are we going to continue on? How are we going to start now trying to find specific molecules that inhibit the pump? And yes, this is only one mechanism, and it's maybe just a very small step. Everybody knows cancer is not just one disease. Cancer is just a variety of different diseases, and every cell is somewhat different. So we try to see, can we do at least one step here? So as I said, people have been looking for 40 years, and actually the NIH by now is convinced that this is an undruggable target, which makes it hard to get it funded. But I think we have some pretty decent results to show that we can actually target it. So our hypothesis was that in the previous studies, people had mostly been looking for, or had been identifying, inhibitors that compete with drug binding, right? So that instead of chemotherapeutic being cycled out, the inhibitor is being cycled out. So that keeps the chemotherapeutic in the cell longer and makes the cells react better to chemotherapy, and so they look good in cell culture, but you need a lot, and all of those clinical trials failed for toxicity reasons. So we thought if we can somehow use the structures, we had all these intermediates of the ATPase motor, and we need ATP hydrolysis to change the conformation, if we selectively block up here and make sure that those compounds do not bind with a drug binding domain, we will have it, oops, no, back. So if we specifically pump out here but not down in the drug binding domain, then we would specifically, then we could find inhibitors that are not pump substrates themselves. So the way we did that was to use very large in silico screens. We screened 5 million drug-like compounds that you can get in databases. We used the Zinc database, and we screened them against the ATPase motor, okay, and then we found a certain number of good predicted binders, and then we screened them against binding to the drug binding domains and subtracted all of those that interacted well with the drug binding domain from our screen. So we started out with 180,000 drug-like molecules that interacted with the ATPase motor. And in the end, after we subtracted those that also interacted with the drug binding domain, we only had like 300 left, and now 120 or so were assayed in my lab. So as a biochemist, I thought let's start with biochemical analysis, right? So we were trying to screen for inhibitors of ATPase, so that's what we did, and we did ATP hydrolysis activities and screened like 45 initial compounds, and we found 4 that inhibited ATP hydrolysis to a degree. So that's what we focused on. I mean, we have to start somewhere, right? So we had, these are the 4 molecules that we found, and you can see they're structurally pretty dissimilar, not too much, not too much similarity here chemically. They docked. 3 of them docked according to a program in the nucleotide binding sites, and the 4th one looked as if it docked in a more allosteric site, not directly interfering with ATP binding. So we tested that. All 4 of them inhibited ATP hydrolysis in a concentration-dependent way. I have a way on how to test for ATP binding. I can calculate ATP binding by using a spin-labeled analog of ATP. And we could show with those, I'm not going to go into the technique, we could show that 3 of those inhibitors inhibited ATP binding to a degree, and those were the same ones that were predicted in the model to interact with the nucleotide binding domain. So that confirmed our hopes that we had a good model. But now we need to see how it works in the cells. Okay, so we found prostate cancer cell line pair, where the open circles here correspond to a naive cancer that has not seen chemotherapy, and the filled circles respond to a cancer that is highly overexpressing that P-glycoprotein. So if you look here, this was really a strike of luck that we found this cell line first. But we had about 1,000-fold concentration window that we could work with here. So that's a really good signal-to-noise ratio. So if you add our compounds to the highly overexpressing P-glycoprotein cell line, the concentration, the toxicity of the chemotherapeutic was pushed right back to the naive cell line. So that was really interesting. That was 3 out of 4 did that. The 4th one, yeah, well, we weren't smart enough, we weren't looking close enough, but this one has a couple hydroxyls here that are actually acidic, so it's probably negatively charged and never gets into the cell. It works well in an isolated protein, but not in cell culture. So we did a lot of assays to confirm that we actually are keeping, or that in the presence of our inhibitors, we keep transport substrates in the cells. So this is an accumulation assay of a PGP substrate. On the left, without inhibitor, it stays dark. That means everything gets pumped out. In the right, with inhibitor, it gets like, it lights up, and that means it stays in. We made some spheroids of the same cancer cells, and could show again that we had uptake of transport substrate in the presence of our inhibitors. We could show that in the presence of inhibitor, those tumors shrink over time, while the one that has no inhibitor stays the same. We kept going, your typical migration assays, again in the presence of inhibitor, and we did it all with different types of chemotherapeutics, but in the presence of inhibitor, there was no cell migration, and in the presence of just a chemotherapeutic, the cells didn't care. We did colony formation assays. I think this was also a different cell line. So I'm going to just go through this one because it's the easiest one to see. So dark purple means that cells are happy and growing. This one means nothing is growing. So in the presence of DMSO, the vehicle, cells are growing. The two different types of chemotherapeutics, the cells are still happy. Our compounds, the cells are happy, but as soon as you have the combinations, there is no cell growth. And we could show that actually none of those compounds were pump substrates. So we did some pump assays for that. And this one is an experiment that I want to show. That's really exciting to me. In this case, we have, let me see, that's the one. We incubated those spheroids with the compound alone, and we let it grow for 72 hours, and it grows nicely. In the presence of chemotherapeutic alone, it grows nicely. In the presence of chemotherapeutic plus inhibitor, okay, you see it here, the red means dead, so the cells are dead. But if you change the experiment and only incubate for 3 hours, okay, after 3 hours, nothing, okay, 3 hours of paclitaxel chemotherapeutic, then you wash it away, and then nothing really happens because the cells are okay. If you do it with a chemotherapeutic N29 for 3 hours, not enough time. But if you take the chemotherapeutic and the inhibitor for 3 hours, then wash the chemotherapeutic away and just continue to incubate with the inhibitor, the cells still die. So it just took 3 hours, and then locking in the inhibitor by continuous exposure to our inhibitor, blocking in the chemotherapeutic by continuous exposure to the inhibitor, the cells died similarly as if we had continuously incubated them with chemotherapeutic. So other cancer cells work the same way, try to be good in time. So this one is with a very overexpressing ovarian cancer cell line. The previous one was prostate. So all of these things work really well. And I think I'm going to skip over this mostly because I want to stay in time. Those are just very initial hits, so they need to be refined. We are looking at KDs in the 10 micromolar range. It's way too high, so we have to make them better. So we again started out with computational methods. We still have to work on it, and we focused on that one compound 29 which as an allosteric inhibitor is a little easier, I think, than having ATP binding inhibitors. There could be too many side effects. So we went to our local organic chemist. Unfortunately, we do not have medicinal chemists in the department or in the school, and asked him how we would synthesize that compound 29. And he gave us a reaction scheme, and then computationally we varied the different parts and came up with a wide variety of variants of 29. So we tested those, and I'm not going to go in detail, but what we found was that a group that went through, that was created by, just variants being made through the computer and then docked the same way, the same subtractive way as in the previous studies, they all did what they were supposed to do. They all were not transport substrates. They all inhibited ATP hydrolysis while the nucleotide binding remained. In the second group, we just did an informed, where the chemist looked at the binding site within the structural model and tried to figure out what he could do to make them better. And for those, 4 out of 5 actually were transport substrates. So that goes to show that that thing just transports just about everything. Slowly going forward, COVID had slowed everything down in my lab a bit, but we also wanted to look at the cousin, the breast cancer resistance protein. We made a breast cancer cell line in the lab that was highly overexpressing the breast cancer resistance protein to 100 nanomolar mitochondrion. We did an initial screen, we found some hits. We did the initial same kind of studies. Are we keeping transport substrates in the presence of the inhibitor? And we have a few really nice hits. The problem was the cell line that we had created was misbehaving. It did stuff that we didn't want it to do. It obviously had accumulated other mutations during the process of while we made it. So we were looking for other cell lines. And this is where Amit comes in, because I was checking in the literature who else has cell lines that overexpress BCRP, and he was the one who responded after I asked him, and so we have been working on this, but his cell line is also overexpressing a little bit too much. If we want to go towards maybe preclinical studies, making mouse models, using those cell lines in mouse models. So what we're doing right now is trying to get cell lines that have overexpression of BCRP, but not to such a high level. We want to be more in a clinically relevant level. So we have several different cell lines that are being made, and we correlate them in their susceptibility to being inhibited, to the window that we can see, and how well they behave, put it that way. So this is where we're going right now. We're going to see if we can get those cell lines so we can go into an in vivo model and see how well those inhibitors fare. I'm worried that they will not. I'm worried that they will not be stable enough, so I'm pretty sure that we do need to do a lot more medicinal chemistry before we have something that works. So I showed most of the people who were involved. So this is just the last couple slides showing SMU. We are a beautiful campus, small private school. We have about 6,000 graduate students, 6,000 undergraduate students. We're trying right now to work on international collaboration so that we can have maybe some graduate students from here or from other countries come, and we'll have to work something out. I think it'll go. Okay, and that's it. And I think I made up it. So, if there's questions.
So, they'd been working on this protein for a long time and been unsuccessful. So you decided to get around that by targeting essentially a different part of the protein, right?
Mm-hmm. And excluding a part of the protein that I think is problematic.
Yes. So, but when there are a lot of ABC transporter proteins, is there a possibility that your molecules could be affecting a number of them because of where you're targeting if it's an invariant portion of the protein?
So, yeah, that's absolutely possible. What we're trying to do right now with the BCRP and PGP, right? So we have some that are specific for PGP, we have some that are specific for BCRP, and we have some that get both, yeah. So, okay, so initially my idea was those pumps are important in the blood-brain barrier as well. So if we knock them all out, and we actually don't have to knock them out because the cancer is only overexpressing one of the pumps, then we could leave the other door open, right, so that stuff can get out of the brain again. But then, like Amit said yesterday, many of them may do more than one, and so my idea was, and it's probably pretty naive, type the cancer, figure out what it's doing, and then target it with a specific inhibitor or with a non-specific inhibitor. Yeah, okay, one thing I haven't really brought up. The inhibitors, the ones that we are looking at, the next screen is, of course, are they toxic to normal cells, right? And they're not, or they're tolerable.
Yeah. So this room is full of chemists.
Yeah, well, if whoever is willing to help me with medicinal chemistry.
Many chemists here, and they were looking to see, what are the possibilities to collaborate in that area. So.
Yeah, I mean that would be fantastic. That's what is missing right now. I need somebody to help me make those things stable, make them better. And
you've seen the reactions also.
And what? Yeah, what?
So I have noticed the reactions also.
Okay.
No, it's okay. We'll discuss later. Yeah, we will discuss later. Yeah.
Okay. Yeah.
There is a question in that.
So thank you so much. It was a nice talk actually. So I'm interested in docking models, basically your MD simulation studies.
Mm-hmm.
So for how long have you performed those studies, MD simulations?
You mean the time frame?
Yeah.
Okay, now I have to not answer this question because I did not do those simulations, but I can find out for you.
Okay.
That was in collaboration.
Yeah, and one more thing. Were you interested in making a specific kind of inhibitors, like because you have shown in one slide, once you treated paclitaxel 29 combination for like 3 hours, then 29 with 69 hours, it was showing like completely,
Same as
cell death, that kind of thing.
Yeah.
Yeah. So, are you really interested in making some small, specific kind of inhibitors?
Well, yeah, so what I think is happening in this case is that it only took 3 hours of incubating with the chemotherapeutic to get the cell loaded to a toxic enough environment, and then we lock it in by continuing to incubate with the inhibitor. It can't get pumped out. So that could potentially affect how chemotherapy is being used in the future, because we would not have to continue to give the highly toxic chemotherapeutics. We could just give it for a short time once, and then keep the inhibitors there and kill the cancer cells.
Last question. So, is there any, a plan for glioblastoma treatment kind of thing with?
I'm really open to anything here. Those are, of course, very important cancers that are hard to get. Yeah.
Okay.
Yeah.
Yeah. Thank you so much.
Open for any collaboration, and especially if I can find medicinal chemists. That is really cool.
Thank you.
Thank you, ma'am. Thank you so much for your words of wisdom. It will be a great pleasure for us to listen to you again in our future events. Now, I would like to request Dr. Piyush Trivedi to present the memento to Dr. Tamanna Narsinghali, the session chair, and Dr. Priya Vogel, the keynote speaker.